My name is Owen Pearce. I am a Senior Research Associate at eGenesis, and today I'm going to be talking about engineering porcine genomes for xenotransplantation, detecting structural variants in engineered genomes using optical genome mapping. eGenesis is a gene editing genome engineering company that is committed to the development of safe and effective human transplantable organs, tissues, and cells to address the global organ crisis. The organ shortage crisis is a huge unmet medical need. As you can see from the chart on the left, every year there are over 100,000 individuals currently on the transplant waiting list, and that far exceeds the number of transplants or donations that we can perform. Here at eGenesis, we believe the porcine donor can be an alternative source of organs for human transplantation. And why are we interested in pigs as donors? Well, there's a number of different advantages, notably the comparable size of porcine organs relative to human organs, similarities in the physiology of these organs, as well as pig farming being a cost-effective and well-established method in today's society. In addition to these advantages, there are a number of challenges that will need to be overcome in the world of xenotransplantation, namely the zoonotic risk or immune rejection between molecular incompatibilities of porcine donors and human recipients, as well as physiological incompatibilities. In order to overcome some of these challenges, eGenesis has developed the eGenesis Genome Engineering and Production, or eGen platform. The eGen platform is a convergence of technologies that we use to produce human-compatible, or HUCO, organs and cells. The process begins with genome engineering performed on primary porcine cells. Once we've genetically modified a cell, it goes through a round of single-cell cloning. These cell clones are expanded and put through a number of different genetic verification tests. And once we've identified the optimal modified cell, that nucleus is inserted into an enucleated oocyte. This oocyte is artificially activated and transferred into a surrogate to begin the development of our porcine donor. This process all occurs at our state-of-the-art cloning and production facilities and has been used to produce porcine donors efficiently and effectively. Once the porcine donor has reached the age of procurement, the HUCO organ or cells are transplanted in partnerships that we have with leading transplant centers around the US. Zooming in a little bit on the genome engineering pipeline we have in the eGen platform, there are three major genetic edits we hope to perform. First, in order to address the zoonotic risk of porcine endogenous retroviruses, we will look to inactivate all copies of porcine endogenous retrovirus in our porcine donors. Porcine endogenous retroviruses are native to the porcine genome, and they have been shown to have the capability of infecting human cells. So we use programmed guide RNA and CRISPR-Cas9 technology that has been exclusively licensed to eGenesis from the lab of Dr. George Church, our scientific co-founder, to inactivate these retroviral elements in the porcine genome. Additionally, to help prevent graft rejection, we perform a triple gene knockout, or 3KO, to improve the compatibility between species. Again, we used a programmed guide RNA and CRISPR-Cas9 technology to target three specific genes in the porcine genome, which encode glycans on the that are expressed on the surface of all porcine cells. Most humans have preformed antibodies against these three glycans, and so by knocking out these genes, we are drastically improving the compatibility between the porcine donor and human recipient. In addition to these genomic knockouts, we will also perform a knock-in or insertion of our human transgene payload. These encodes a number of human genes that we use to address key mechanisms of graft rejection, namely the innate immunity incompatibilities, coagulation, adaptive immunity, and complement regulation of the recipient immune system against our porcine donor. So these human transgenes are inserted as a payload into an identified genomic safe harbor in the porcine genome, where they are stably and ubiquitously expressed in all cells of our porcine donor. Our recent publication has demonstrated a proof of principle study and helps show the clinical potential of xenotransplantation. So our article released in Nature at the back end of last year, titled Design and Testing of a Humanized Porcine Donor for Xenotransplantation, has demonstrated that renal grafts from engineered porcine donors show long-term survival in NHP models up to 758 days. As you can see from the survival curve on the right, when the 7 transgene construct is introduced into our porcine genome, The survivability of these renal grafts is drastically improved when compared to the 3KO and retroviral inactivated donor alone. So as we approach this key clinical threshold as a company, there is a need for us and xenotransplantation as a field in general to develop in-depth characterization of our engineered genomes to monitor intended and unintended alterations. These alterations may be small indels that occur at the site of our Cas9 engineering, or large structural variants. These structural variants can be classified into two main groups, either intentional genomic alterations, and these are edits that are intended as part of our engineering platform that I've described previously, namely retroviral inactivation, the xenoantigen or 3KO knockout, and the human transgene payload insertion. The other classification of genomic alterations are UGAs, or unintentional genomic alterations, and this can include anything from plasmid backbone integration into the porcine genome, concatameric payload insertions, retrotransposable element mobilizations, or any other broad structural variants. We have shown that our porcine donors containing 69 genomic edits have a normal porcine karyotype result of 38XX, but what do we see if we look closer? For example, G-band karyotyping only shows megabase pair scale resolution and would miss instances such as this, wherein an FDA hold was put on this product's attempt to generate a hornless cattle breed when it was detected that the template plasmid had integrated into the germline of these gene-edited cattle. And so in order to avoid situations such as this, we need technologies that exist to look at the genome at a smaller resolution than traditional G-band karyotyping. So here at eGenesis, we use a number of different technologies for monitoring genomic alterations. First among these is optical genome mapping, and that is supplemented alongside a variety of next-generational sequencing techniques. The process begins with running cell lines of interest on the Bionano Sapphire instrument, where we will use 750 nanograms of ultra-high molecular weight DNA, run it on the Sapphire to generate high-level locus info and identification of structural variants in our assembled genomes. We will then take some of the high molecular weight DNA that we have generated using the Bionano nucleic extraction kit and sequence this DNA on our Oxford Nanopore PromethION 24. Running it on this system helps us generate sequence and genome scaffolding information that we can use to characterize these structural variants as either intentional or unintentional. For key cell lines of interest, we will then use the PacBio Sequel2 instrument to generate additional sequencing information with high quality and high fidelity base calling information. Before we could use Bionano optical genome mapping for our characterization of genomic alterations, we first had to optimize the wet bench portion of the Bionano optical genome mapping process for using our main cell type of interest, which is namely primary porcine ear punch derived cells. As you can see from the charts on the left, we used a number of different starting materials for this process to identify the best possible starting material, and we have found that freshly cultured cells or cells taken from viable cryovials, as opposed to snap frozen cell pellets, generates the best quality DNA. As you can see, when you go from N50 greater than 20 to N50 greater than 150, pellets show a much more fragmented quality of the DNA as compared to the fresh and cryopreserved cells. In addition to these sample input optimization, Bionano has also recently rolled out their new generation of chemistry named G2. And using this kit, we can generate even longer molecules and even greater quality maps. So we are now generating optical genome maps with N50s approaching or greater than 400 kb. In addition, you can see from the two graphs on the bottom right, the labeling reaction is far more robust and reproducible, which gives us further confidence in the genomic assemblies we are generating using the Bionano system. Circling back now to the paper I mentioned previously, I will talk about how we characterize the intentional genomic alterations that we used to generate our transgenic xenotransplantation renal donors. So starting with wild-type cells, we used CRISPR-Cas9 NHEJ to perform the triple knockout of our xenoantigens, and CRISPR-Cas9 HDR to insert a landing pad at our proposed genomic safe harbor. We then used recombinase-mediated cassette exchange to introduce our human transgene payload. In this case, it is our payload 15S, which encodes 7 human transgenes and is about 21 kilobase pairs in size. Lastly, cells from these pigs were used in a further round of engineering to introduce the retroviral inactivation edit. As you can see from the optical genome mapping data shown in the table below, during the landing pad knock-in, we were able to detect a 2.5 kb homozygous insertion at our genomic safe harbor, which represents our landing pad, with no presence of landing pad template backbone found in our porcine genome. Following the cassette exchange, we were able to have a hemizygous genome genotype at this loci, with 1 copy of the landing pad remaining and 1 copy exchanged for our 21 kb payload 15S. As was the case with the landing pad, we had no presence of the plasmid backbone in the sample. Following the retroviral inactivation step, the genotype of this loci was unchanged, suggesting we have a stable and robust edit, given that this genotype did not change during a round of engineering or somatic cell cloning. Next generation sequencing technology was used to confirm these findings, and it all agreed with the optical genome mapping data. Here is just showing some of the optical genome mapping tracks that we used in showing how the unbiased and haplotype aware method of the optical genome mapping assemblies can help reveal the accuracy of our genomic integration events. So as you can see in our reference genome, there are 2 DLE1 sites marked with the black arrows. And as we introduced our landing pad, we see a shift in the location of these DLE1 sites, supporting a 2.5 kb insertion. We also introduced a novel DLE1 site shown with the yellow arrow, which is encoded in our landing pad. After we performed the cassette exchange, you can see a further shift in these DLE1 sites relative to the reference genome, and that is supporting of a 21 kb insertion, which is the approximate size of our payload. We've also introduced 4 novel DLE1 sites that we have identified as being encoded in our payload. You can also observe the 3KO 7 transgene RI genotype is identical to that of the 3KO 7 transgene at this loci, suggesting again the stability and robustness of our edits. Next, we hope to characterize our unintentional genomic alterations. So as you progress from the left to right of this table, our wild type cells to the 3KO genotype, we did not detect any structural variants. From the 3KO to the 3KO 7 transgene, we detected 3 insertion events. And then from the 3KO 7 transgene to the retrovirally inactivated genotype, we have an additional insertion event with 2 deletion events detected. We will then look at these loci and structural variants identified through optical genome mapping in our NGS datasets to help characterize them. And we have demonstrated that all 4 insertion events were found to be line retrotransposons that have inserted at sites independent from our anticipated on-target CRISPR sites, in addition to our predicted off-target sites. The deletion events consisted of an intergenic region being deleted, as well as the deletion of an entire copy of the porcine endogenous retrovirus. Here is some optical genome mapping data supporting one of these UGA calls. As you can see, the top down 7 tracks all align directly to the reference genome as compared to the final track of the 3KO 7 transgene RI, which has a shift which supports the 8 kb insertion. If we were to look at this loci with next generation sequencing technology, again, you see the agreement between all 3 assemblies and the reference genome, with the difference being the 3KO 7 transgene RI has half the reads supporting the reference, the other half demonstrating an 8 kb insertion. This is supportive of the heterozygous insertion called from optical genome mapping. And if you were to blast this sequence, you would find that it aligns to line element insertion into an intronic region of our porcine genome. So to conclude, we've established that Bionano optical genome mapping technology enables an unbiased and subchromosome scale interrogation of engineered porcine genomes. We've demonstrated that it's superior in resolution to traditional G-band karyotyping methods. In addition, Bionano optical genome mapping has provided us with genome-level clarity of structural variants, as well as assistance in the identification of these structural variants. Orthogonally, we can use NGS-based approaches to enable base pair scale resolution of these structural variants and help us classify them as intentional or unintentional alterations in the genome. We've also shown that lines are active in pig genomes, with thousands of L1 elements previously identified in pigs. Also recently, line insertions have been reported to occur at sites of Cas9 editing, both at on-target and predicted off-target sites in transformed cell lines like HEC293T, HeLa, and U2OS. However, we could not detect line insertions at any of these sites, either on-target or off-target, for our predicted genomic edit loci. With that, I would like to acknowledge eGenesis colleagues past and present who have helped contribute to this work, namely our molecular biology and development teams, our nuclear donor cell engineering team, our computational biology team, as well as our production and cloning teams. I'd also like to acknowledge everyone at Bionano who has helped support us over the last 2.5 years, in addition to Dr. Catherine Brownstein and the Boston Children's Hospital Molecular Genomics Core, who helped us running pilot experiments prior to the initiation of the Sapphire here at eGenesis. With that, I'd like to thank you for your time, and I hope you enjoy the remainder of the symposium. Thank you.